Today we will be discussing the analysis of Hughes' 2019 coral reef system. This project was done by Andrew Nabarra, Michael Houston, and Michael Dimash. Coral reefs are assessed by the abundance of organisms such as coral, algae, herbivores, and predators in marine systems. Overfishing has caused these organisms to drastically shift between a high and low grazing rate, causing recruitment from surrounding systems to be necessary. This project will discuss a coral reef system and look at the effects of recruiting coral and microalgae into the system and how different grazing rates and recruitment rates affect the system. How these aspects affect the population density of coral, algal turf, and microalgae will be the primary focus within this coral reef model analysis.
So the Hughes model stems from the model introduced by Mumby in 2007, which shows the competition between coral and macroalgae for recruitment onto algal turfs. This competition is denoted by these two differential equations. The first, coral, which is a function of recruitment onto algal turf minus the mortality of the coral and minus the overgrowth of macroalgae onto the coral. The second represents macroalgae, which is again a function of its overgrowth onto coral, grazing by herbivores, and it's spreading over algal turfs. This model makes the assumption that the seabed is covered completely by these three factors, and that each of their respective fraction of coverage can be found mathematically by manipulating the equation C plus M plus T equals 1. Coral being C, macroalgae being M, and algal turfs being T. Since the coral reef is not an isolated environment, potential immigration of surrounding coral and macroalgae is bound to occur. Toby Elmhurst, Sean Connolly, and Terry Hughes extend the Mumby model by accounting for the recruitment of these migrants. They conserve the assumption that the seabed is entirely covered by these three factors and include an additional term to each original equation for this recruitment. They assume that the external coral and macroalgae only recruit to algal turfs and that there's no overgrowth of external macroalgae onto the native coral. This leaves us with the rates zeta and epsilon representing external coral and macroalgae recruitment respectively.
Within the models analyzing the stable states of coral reef systems, the following parameters were used to produce varying system models. The overgrowth rate of coral on turf is 0.55. The death rate of coral is 0.24. The overgrowth of macroalgae on coral is 0.1. The overgrowth of macroalgae on turf is 0.77. And our varying grazing rates are 0.22, 0.285, and 0.370. For the Hughes 2009 models of the coral system, the following parameters were used to produce varying system models. The rate of the external recruitment on turf is either 0 or 0.005, and the rate of external macroalgae recruitment on turf is either 0 or 0.005.
On this slide, models produced using the differential equations for the Mumby models can be seen. On the left side, the first vector field displays a model with a grazing rate of 0.2. And in this model, a trend towards macroalgae dominance and coral death can be seen, where macroalgae thrives, turf takes up the remaining space, and coral dies out. In the second vector field, a grazing rate of 0.285 was used to produce this model. And the stable states of the system depend on the initial values of the population. Depending on the initial values of the population, a trend towards coral dominance and macroalgae death would be seen, or a trend towards macroalgae dominance or coral death would be seen, with both models having the remaining population taken up by turf. In the third model on the right side, a grazing rate of 0.37 was used, and the model shows a system of coral dominance within the system and macroalgae death, with the remaining portion of the population taken up by turf. These three models do make sense when thinking about the system, because at higher grazing rates, the macroalgae should be seen to die out and would allow for coral to thrive within the system. On this slide, the previous vector fields can be visualized in terms of their population densities and how the populations will change over time. So on the first model with a grazing rate of 0.2, you can see that the macroalgae dominate the system over time, with the coral dying out and the turf taking up the remaining portion of the system. In the second model, you can see that the coral population thrives and the macroalgae dies out, with the turf taking up the remaining portion of the population. But with this model, depending on the initial values you use, you could see a system where the macroalgae dominates and the coral dies out. So that would be the opposite from what's shown in the graph. And then for the final graph on the right side, a grazing rate of 0.37 was used, the macroalgae dies out and the coral dominates, with the turf taking out the remaining portion of the system, and the population sizes you have, the coral will always dominate and the macroalgae will always die out. On this slide, the vector fields for the Hughes models of the coral system can be seen, with coral and macroalgae recruitment rate on a turf being displayed on the left side and grazing rates of macroalgae being displayed above the vector fields. In the first row, the graphs with the recruitment rate of 0 for both coral and macroalgae under turf can be seen, displaying the same trend as those seen in the Mumby, which makes sense because there is no change to the system. There is no algae or coral recruitment onto the turf. In the second row, coral recruitment onto turf is introduced, and slight changes within the vector fields can be seen, but the overall trend shows no difference in terms of the final population densities. In the final row, macroalgae recruitment onto turf is displayed, and again, there are slight differences in the vector fields, but the overall trend shows no change when compared to the initial Mumby models. On this slide, the vector fields above for the Hughes models can be visualized with the same rates of coral and macroalgae recruitment onto turf and the same grazing rates of macroalgae. As seen in each row, there is no significant change when coral and macroalgae recruitment onto turf is introduced. So in the first column, you can see that the macroalgae always dominates the system and the coral always dies out. In the second column, you can see that the coral always dominates the system and the macroalgae always dies out, with the remaining population being composed of turf. And depending on the initial values you choose for the population, the opposite effect is seen. And in the final column, you can see that coral always dominates this system and macroalgae always dies out, no matter where the initial values for the population are.
In conclusion, the recruitment of coral and microalgae on the turf did not make a significant difference when compared to the Mumby models. A stable population where coral and microalgae both survived within the system was not found at a grazing rate of 0.285, the rate found in the Hughes model. This grazing rate was seen to display different stable states depending on the initial population values, where one or the other population dies out. Grazing rate of microalgae was a primary cause for the change in the system, but recruitment of coral and microalgae did not seem to create a significant change in the system.